All right. So here we go with mechanisms of evolution. Oh, that's not my pen. There it is. Part 2. Finally, the completion. All right. So we've already talked about mutations and gene flow. And so now we're going to talk about a couple of more ways that we can see microevolution. Okay. Evolution at the population level. Allele frequencies can change, again, via mutation, via gene flow, movement of alleles in and out. But allele frequencies can also change just by chance. Some random event, a lot of times associated with a natural disaster, can have an effect on allele frequencies in small populations. So, chance events. So again, if bad luck prevents some members of a population from reproducing, their alleles are removed from the gene pool. Now, what kind of bad luck are we talking about? Examples of bad luck. You have seeds that fall into a pond or onto a parking lot. Okay, their parking lot doesn't have a lot of dirt, so they're probably not going to grow. Flowers destroyed by wildfire before they were pollinated. They didn't have the chance to pass their alleles on. Any organism killed by a flood or a forest fire prior to reproducing, that is certainly bad luck. Okay, so these, when these allele frequencies change by chance, completely random chance, this is called genetic drift. Okay, so genetic drift doesn't really have an effect on large populations. It only has an effect on kind of smaller populations. Okay, all right. So genetic drift is where chance events cause change in allele frequencies. It again has a much more significant effect on smaller populations. And in smaller populations, genetic drift can even result in the complete loss of an allele in just a few generations, even if it is more frequent. And we're going to look at two ways that this can occur. But first, we're going to talk about like a little bit what we mean by you can completely lose an allele. So if you look here, the frequency in this population, if you counted up all the dominant alleles, all the recessive alleles, the frequency of the dominant allele is 50%. So you see it half the time. The frequency of the recessive allele is 50%, half the time. Okay. So again, let's say that only 2 individuals in each generation are chosen to breed. So let's say we choose a heterozygote and a homozygous recessive. Okay. Now, just doing that alone in one generation affects significantly the allele frequencies. If you count them up again, the frequency of the dominant allele will be 25%. The frequency of the recessive allele is now 75%. And then again, if we just randomly choose 2 individuals, if we choose 2 homozygous recessive, that basically in the next generation can completely eliminate the dominant allele. And again, just chance, depending on who you choose, it could have easily gone the other way. Okay, and that's what genetic drift is like. So again, large populations, this really doesn't have much of an effect because there's too many instances where it can be preserved. But in small populations, okay, you can have has a much more significant effect. All right, so there's 2 types of genetic drift. One is a population bottleneck, or what we call the bottleneck effect, and the other one is the founder effect. Okay, so these are 2 types of genetic drift. So first we're going to look at bottleneck effect. So in the bottleneck effect, a population is drastically reduced by a national catastrophe or overhunting. Okay, so something happens, forest fire, or complete overhunting of an organism for meat or trophy. So you reduce the population to very few individuals, and then they're the only ones who have genes to contribute to the next generation. Okay. And again, this can very rapidly change the allele frequencies. Okay. So there's a huge reduction in the number of individuals in the population. Okay. An example of bottleneck effect with the northern elephant seal. They were hunted almost to extinction. In the 1800s, there were only 20 individuals left by the 1890s. Okay. So we've drastically reduced the population to a very small number. So a hunting ban allowed the population to increase to about 30,000. But because there was such a small number of individuals to start off with, there wasn't a large number of alleles left in the gene pool. So here, we're going to use our bottleneck to show how this works and why it's called a bottleneck. So you have an original gene pool that has, let's see how many alleles. There's yellow, there's blue, there's green, and there's red. So there's 4 alleles. So let's say we randomly turn the bottle upside down and shake out 20 beads. Okay. And just by total random chance, only blue and yellow are shaken out, and those are the ones you have left. So now, if the population rebuilds itself, okay, there's only those 2 alleles. 2 other alleles have been completely eliminated, okay? So the the gene pool, the new gene pool, isn't representative of the original gene pool, okay, original. All right, and there's our friends, the elephant seal. Okay, now when this happens, it really can affect the genetic diversity, obviously, if we're completely eliminating alleles. Biochemical analysis shows that present-day northern elephant seals are almost genetically identical. Despite their numbers, they lack genetic variation, and this is going to affect their ability to evolve if their environmental circumstances change. We better hope that the elephant seals start getting mutations like crazy cakes. Okay, so that's bottleneck. Next, we're going to look at the founder effect. The founder effect, again, the end product is similar. You have a small population that has a gene pool that's not representative of the original gene pool. But the founder effect occurs when a small number of individuals leave a population and establish a new isolated population. And if you think about, like our founding fathers, right? So they, kind of packed up the wagons on the East Coast and trucked out west and settled down and formed new colonies. And this is kind of where they got the name, the founder effect. So a small number of individuals left the original population and founded a new population elsewhere. Okay. And again, just whoever left with the small group, their gene pool is the new gene pool of the population. And again, the allele frequencies of the founders may differ from that of the original population. So again, over time, the new population will exhibit frequencies that are different from the original population. Okay, so those were the examples of genetic drift. Okay, our fourth thing that can affect microevolution is mating. Okay, mating within a population is almost never random. Okay, and when you have non-random mating, it can play a significant role in evolution because for a number of reasons, but basically it kind of keeps certain alleles more popular while eliminating or decreasing the frequency of other alleles. All right. Examples of non-random mating. Some species lack mobility and tend to stay near their birthplace. Okay. So offspring have parents living in the same area. They may even be related. Sexual reproduction between relatives is called inbreeding. We saw this a little bit when we talked about genetics, and we talked about the royal families and that kind of stuff. Consanguineous matings, inbreeding, and when this happens, again, because relatives are genetically similar, inbreeding tends to increase the number of individuals that inherit the same alleles. Okay. And remember, we saw that with the hemophilia and some other disorders. It enriches certain alleles, and a lot of times it enriches alleles for recessive disorders. Okay, so increase in homozygotes can have harmful effects, and we talked about that when we talked about our genetic disorders. So inbreeding is one example of non-random mating. There's also another example of non-random mating called assortative mating. And basically, this can happen if individuals have preferences or biases that influence their mate of choice. So our good friend, the snow goose, comes in two colors, white and blue-gray. And the mate choice is based on color. It's not random. Okay, so white geese mate with white geese, blue-gray geese mate with blue-gray geese. Okay, and again, this helps to kind of isolate and reduce the genetic diversity in the gene pool. Okay, so neither of these, of course, by themselves, neither inbreeding or assortative mating, will alter allele frequencies, but it is going to change the distribution of genotypes and therefore phenotypes in the population. So it'll keep things, it actually keeps things more similar, and this doesn't generate enough genetic diversity. Okay. Okay. Because it's not generating genetic diversity, and again, all genotypes are not equally beneficial. So in a hypothetical equilibrium population, remember, we're still kind of circling back to our Hardy-Weinberg. So if you have a hypothetical population, all genotypes will survive and reproduce well, and no genotype will have an advantage. And there are instances in nature where certain genotypes do have advantage. All right. The last thing we're going to look at, reason number 5, is natural selection. Okay. Again, all genotypes are not equally beneficial. So sometimes you have certain genes that are going to provide a benefit, that are going to provide a selective advantage. Okay. Natural selection is the process by which these individuals with traits that help them survive and reproduce leave more offspring than individuals without those traits. Okay. And I think that's it. Okay, we've talked about natural selection a ton. So that's all. And now you know about all that stuff.